Hello, everyone, and welcome to our online lecture series for genetics. Today, we're continuing on with this quest that we've set on of understanding three-point test crosses and genetic recombination in combination with topics of chromosomal assortment. This is our continuation of our heaviest math part of the lecture, where we're going to begin today by going over another chi-squared problem, as well as another three-point test cross where order is unknown and distances are unknown. And then from there, we're going to take a little bit more of a biological spin, making sure that you understand the math is very important, but we're also going to try to understand some of the molecular bases for these markers and linkage mapping and why it all matters in the end. So let's start out with that chi-squared problem. This time, rather than assuming that the distance between two loci, B and D, is 50 centimorgans, or that they exist on separate chromosomes, we're going to set an expectation that I'm going to tell you that these two loci are 12 map units apart. In order to determine whether I'm lying, we have to go through some of the data needed to ascertain whether that statement is true or false. So the first thing we're going to do is we're going to have an individual that is heterozygous at both of these loci, B and D. Now we don't know the order of these, other than they're adjacent to one another, we don't know the locations of these, but we do know that they exist in a single individual and that there are two different alleles for them. We're going to be indicating the alleles as ones and zeros, just to make sure that you don't try to superimpose dominant recessive or any other kind of genetic assumption about these. We can just tell that they're different. In order to understand the genetic distance between these two loci, we have to perform a test cross. So what's a test cross, other than the genotype behind this gray window? Well, a test cross is where you cross it against an individual all of one genotype, an individual that is all zeros. Because what's going to happen when you cross the individual that is heterozygous against the individual that is homozygous of all one sort? Remember, not dominant, not recessive, but rather just one form. Well, you're going to get a number of different genotypes out. If we're doing a Punnett square, we would assume that all four of these would be produced in equal ratios. But because I'm suggesting that these are linked, we're going to try to see if we can infer something about which versions of these alleles are linked in the parent. Because remember, just because we write them 1 and 0, 1 and 0, that does not mean that 1 is found on the same chromosome as 1, and 0 might not be found on the same chromosome as 0. We have to look at segregation ratios of the progeny. So take a moment here and first write out all four possible genotypes of the progeny produced by a heterozygote or dihybrid crossed against a tester. I'll go through it first. We can have a genotype that matches the hybrid parent with B1, B0, D1, D0. This would be produced from a very particular gamete. So I'm also going to indicate, the gamete that would produce this cross. So we'll have a little gamete indicator, meaning what was the genotype of the gamete donated from this parent to each of these crosses. Well, if this individual got the 1 and the 1, the only way he got 1s were from the hybrid parent. We'll do the same for all the rest of these. I'd of course encourage you to do this before watching this video, or at least give it a pause right now and try to fill in the genotypes yourself. Our next genotype could be all zeros. Following that, zeros with a 1. And following that, literally all zeros. In this case, we would have a B1 D0, a B0 D1, and a B0 D0. So there we have the relationship between the progeny produced and the gamete donated by the heterozygous parent. Now we get to the ever important information about progeny numbers. So for this cross, I will provide the following numbers. We see that there are 400 B1, B0, D1, D0. That there are 1,850 B1, B0, D0, D0. 1,930 B0, B0, D1, D0. And 320 B0, B0, D0, D0. From here, you should be able to infer what the linked haplotypes are. And what do I mean exactly by that? Is B1 found on the same chromosome as D1 or D0 in this hybrid parent? What are our linked haplotypes here? Well, the more frequent are going to be indicative of our parental types. The less frequent should be our recombinants. We see that B1 and D0 and B0 and D1 have the highest frequencies in our gamete pool for our hybrid parent, with the recombinants of B1, D1 and B0, D0 being our less frequent. So in this case, this is our parental, with our other 1, 1 and 0, 0 being our recombinants. So this is one way in which these situations can vary, and I'd encourage you to not make assumptions about linked haplotypes based on numbers, letters, dominant, recessive. So next up, we have to plug this into our chi-squared. Our chi-squared formula again, observed minus expected squared divided by the expected value. And we're going to sum across our different classes, our parental and our recombinant types. So into this box of genotypes, I will write the sum total of our parental and the sum total of our recombinant. Because ultimately the B's and D's and 1s and 0s don't matter, we're just looking for the different types in terms of parentage. Now, our observed number of parental types is going to be the sum of 1850 plus 1930. That value, if I've done my math correctly, is 3,780. Our sum total of our recombinants is 400 plus 720, plus 320, giving you the total value of 720, right? How do we derive our expected values? Pause for effect. Hopefully you took a moment to speak to your electronic device and tell me exactly how I would get those expected values. If you said something along the lines of you have to use the map units, then you're in the right ballpark. Well, our expected value would be our sample size times our map units, represented as a decimal. This will get the expected fraction that we would see if it were true that these two loci, B and D, were 12 map units apart. So, with our total number of progeny, how many did we have actually? 4,500. We can see that our expected values should be 4,500 times 1 minus 0.12, or 0.88, giving us an expected number of 3,960 parental haplotypes. For our recombinant types, we have 4,500 times 0.12, giving us 540 in terms of our recombinant types. That would be expected if these two loci were truly 12 map units apart. Now, what's your gut feeling on this? I mean, 4,500 is a pretty big number. This looks to be a discrepancy of like, I don't know, 200, close to 200, between 200 and 150. Whereas this appears to be a discrepancy of, not dissimilar, another discrepancy of something like 200. How will this carry out into our chi-squared? Well, let's do the math. For our parental, we set up our observed 3,780 minus our expected 3,960. Square that value. Divide by the expected value again, the expected being 3,960. And then go ahead and plug that into your calculator. This will get your chi-squared statistic simply for your parental. Well, for your parental, you should see that the chi-squared value is 8.18 or 8.2. Just for a sanity check, of course, you're not going to forget to go and do your recombinant type. So if the parental's done, we have to make sure to remember the recombinants. So we'll set that up. The observed being 720 minus the expected 540. Squaring divided by 540. And then we'll have to add those two values together. But even before we find out what this solution is for our recombinants, we can take a look at the chi-squared table. Well, if we have 1 degree of freedom, which we do, because you can only vary on your parental or recombinant genotypes, what is our suggested p-value here? Well, with one of the values, 8.2, we see that we are in between 6 and 10 for our chi-squared value, which has a value of less than 1% likelihood of this happening if these were truly 12 map units apart. So from this single value, we can already reject, you already know that this is not likely to be 12 map units apart. It's not by random chance that you're deriving these ratios of progeny if these two loci were truly 12 map units apart. It's going to become quite cartoonish when you look at the value derived from our recombinants. The chi-squared value from our recombinants is 60. So our total chi-squared value is 68.2. What's the likelihood that we get 68.2 from a table where we see these two loci 12 map units apart? Well, it's going to be far in excess of 0.1% of the time. Now, if something occurs 0.1% of the time, or I could be lying to you about how far apart these two are, and you are a betting individual, I'd hope that you take the bet to say that these are not actually 12 map units apart. They're a different distance. And that's what we would do. This should lead you to reject the null. Now keep in mind, you should be able to perform these types of problems as well as write some on your own. It's important to go forwards and backwards when problem solving because that shows that you truly understand the ins and the outs of our chi-squared genetic distance problems. All right, so next up, we're going to move on to our problem of unknown order and distance. Oh, and I forgot to move this back down here. Let's go ahead and just scoot ourselves a little table here for some clarity, make sure that we can keep track of which genotypes are which. But we've got 3 loci back to B and D with a third locus G here, where we don't know the order of these, so don't assume that I write B, D, then G, that D's in the middle with B and G on the sides. If you go ahead and do that, then you're assuming things, and assuming things makes a bleep out of you and me. Doesn't work out well for us. you have to be able to derive the distance, the order and the distance based on the frequencies of sperm created here. So we're going to skip the step of going to our progeny, because keep in mind, genotyping progeny is not dissimilar from genotyping sperm. After all, we did just isolate what were the gametes that were produced to identify the parental and recombinants here. It just happens to be more expensive to do something where you're genotyping sperm than genotyping progeny, particularly if the progeny can manifest the phenotype. So you have both situations in real biology labs. But here we're going to be looking at sperm, meaning that all of our genotypes are going to be haploid. So we've got our single letter per locus, and then we've got our trihybrid. Now in this case, we knew that we are being told that the parents were true breeding. So finding out the linkage between which alleles are connected to which, is it B1 with D1 and G1? Well, once you're told that the parents are true breeding, all ones or zeros, it's easy to assign them to one class or the other. So, at least in this example, we'll keep this straightforward in this regard. Our parental types will therefore be wherever we see ones connected, and our recombinant types will be where we see zeros and ones connected. So let's go ahead and do what we've done before by assigning parental types to each of these sperm. If I look at B1, D1, G1, and I look at the B-D relationships, well, I see both ones. That means I see a parental type. Go ahead and fill this table out. It will be different than the table from before, simply because the list of the order of the alleles here is different. Hopefully you paused and did that. I'll go ahead and fill it out now. D and G have ones, B and G have ones as well. This is parental between B and D, but not between B, D and G, or B and G. Next one is parental between B and G. Next one is parental between D and G, nothing else, both having zeros. After that, we've got parental between D and G again, parental between D and G, B and G, sorry, not D and G, and then parental between B and D, and then our third row, our last one here is parental for all three types because they're all zeros. Conversely, we'll go in and fill in the recombinants, just double checking. D and G, we've got a 1 and 0. B and G, we've got a 1 and 0. B and D, 1 and 0. D and G, 1 and 0. Yep, 1 and 0. 1 and 0. 1 0. 1 0. 1 0. 1 0. 1 and 0. And 1 and 0. Then you're going to go through and make sure that you've got 4 parental and 4 recombinant types in each column. 1, 2, 3, 4 parentals. 1, 2, 3, 4 parentals. 1, 2, 3, 4 parentals. 4 recombinants. 4 recombinants. 4 recombinants. Woohoo! We can assign things correctly. I hesitate to tell you to speed through these things because these problems can be really complicated, and when you're doing a bunch of them, it's easy to kind of overlook things. I've even seen other, and kind of drawing inspiration to see how other folks online teach these kind of things through an asynchronous format. I've seen numerous professors who are parts of teaching teams make errors when assigning parental and recombinant types and forget things and forget rows, right? Just forget to assign a complete row and then your count is off for all of your gametes. So I would encourage you to really be very careful and not skip steps. Establish a protocol that you follow. I've got mine. I write out the different gametes, the different genotypes that are there. I assign them parental or recombinant types. Even before I look at the actual number of progeny created. So let's go ahead and do that now. For our progeny created, we see 532 of this parental type. Again, this parental type, you should expect to see a lot of them. We've got 44 of our, oh, this is kind of hard to see pink. Let's go ahead and change this. I apologize. Let's go with, how does green look on this surface? 532. Yep, okay, that looks very better. Sorry about that. 532, 44, 98, 6 of these, 6 of these. Immediately, snap my fingers. What do you notice when you see 6? That should be where your double recombinants are. They look much, much less frequent than all of our other classes. Rounding that out, making sure that nothing goes below 6, we see 120 here, 45 here, and 558 of our complete parentals. All right, so this should hopefully ring a bell. If you've, I'd encourage you to first solve this by hand rather than immediately going to your spreadsheet or another computational tool. But I'd, of course, encourage solving it both by hand as well as plugging it into a spreadsheet and letting a computer do all of your calculations for you. It's a good sanity check. But we can go ahead and go through and add together all of our values. If you sum together all of the total progeny, you will see that the sample size is 1,409 total progeny. Now we're going to create a sum for each of our recombinants. How many recombinants between B and D do we have? Well, in order to do that, we're going to sum together these 4 values, 98, 6, 6, and 120. And do make sure you double check your math because I definitely just messed up. You may have noticed the pause in the video here, slightly different frame. But we see that 98, 6, 6, and 120 adds up to 230. Going on to our next, we see 44 plus 98 plus 120 plus 45, giving us a total of 307. And then last but not least, 44, 6, 6, and 45, value of 101. So those are a total number of recombinants. Now, in order to calculate the map units between these, or the genetic distance between each of these pairs of loci, we're going to need to divide by the sample size. And then to convert to map units, you can multiply by 100 because of it being a percentage. So 101 divided by 1409 gives us our distance between B and G of 7.2 map units, rounding. Between D and G, very clearly, our farthest apart loci, D and G are 21.8 map units apart. And then last but not least, B and D are 16.3 map units apart. So, with these values, we should be able to plot the order of the loci on a chromosome. Your answer for this should show which of these two are farthest apart. We see that D and G are farthest apart. Does not really matter which one goes left to right here unless you're actually scaling this portion of it, which I typically don't. That's a little bit more excess detail than perhaps is necessary. But D and G are furthest apart with B in the middle. The distance between B and D, 16.3 map units. And the distance between B and G are 7.2 map units, with the overall distance across these loci being 21.8. You'll notice, again, generally additive, but not quite perfect, right? Not quite exact. This again is because we're going to have to calculate our coefficient of coincidence, as well as overall interference. So, dig back through your notes. How do we calculate our coefficient of coincidence for this problem? Well, we're going to need to calculate an observed number of double recombinants and divide that by our expected number of double recombinants or double crossovers. Our DCOs, double crossover events, are the ones where we have the least of them, and just to make sure, they should be recombinants between B and D and B and G, but not between D and G. So we again see parental types in D and G, but then recombinants flanking either side, meaning that this has hopped off and hopped back on in two different locations between our haplotypes. Our observed number of double recombinants is 6 plus 6, or 12. And then our expected number of double recombinants is going to be our distances between the loci multiplied by the sample size. So our distances were 0.072 for our 7 map units times our 16.3, or 0.163 map units, and again multiply that by our sample size of 1409, and you will get a value of 16.5, meaning that we expected more double recombinants than we observed. That should be the way that most of these problems are working out, right? Interference means that you're going to have less double recombinants than you'd expect. Is it possible by some fluke that you have more double recombinants than you'd expect and we see interference being, or coefficient of coincidence being a larger than one number? You could, stochasticity is possible. Keep in mind that the smaller the sample size, the more likely something like that is to occur. But in reality, know that interference, the lowest it can be, is 0, meaning that there is no physical abrasion or physical disruption between these two loci. In this case, the value is not 0, it's 16.5, or 12 divided by 16.5, or a COC of 0.73, with the interference being calculated as 1 minus the COC, or 0.27. So close to 30% interference between our B and G and D loci. Whew! All right. I think that'll wrap it up as far as example problems for triple crossovers. I won't include any more in lectures, nor will I go over chi-squared mapping anymore. You're, of course, welcome to go back and revisit this material. But you'll also have plenty of practice in our problem sets. Now I want to shift our discussion away from these 3-point test crosses in chi-squared back to our realm of biology. Because when we talk about mapping these loci, we need to understand the markers or the information we use in order to determine what identifies one allele from another. Markers are going to be highly beneficial to use, these being molecular markers, simply because many phenotypes are very, very difficult to observe. Imagine how many more things are going to be revealed when you look at the DNA of an individual rather than how they look. How many different shades of brown hair color or lengths of fingertips, and how many alleles underlie each of those various different traits. It can be really difficult to try to quantify things simply by how they look because our brain can't observe all the true small differences between them. So we're much better off by looking at our molecular markers. These molecular markers can help distinguish between different alleles. As illustrated in this graphic here, when looking at our B1 and B0 allelotypes, we'll see that most of the sequence is similar between them. So if we have 7 letters here, the ATG and the TAG are the same, with one single nucleotide polymorphism, or SNP, held indifferent between the two of them. You can also see this in terms of your molecular patterning. I thought this image from Wikipedia was pretty neat. We can see the different shapes for GC and AT. This will also serve as a reminder that the purines have 2 rings. You'll notice the 2 rings on, or the 2 dots on A's and G's, whereas the pyrimidines, C's and T's, have 1 ring. And then you can identify where there are differences in the DNA. We see that there's a SNP here where the top strand, 1, has a GC and the bottom strand, 2, has an AT. Now these SNPs are worth talking about. So let's go ahead and dive into these. We've talked about them a fair bit. Hopefully going to illuminate some different aspects of SNP biology here. So a SNP is a single nucleotide polymorphism, and it must segregate within a population. What this means is that in order to be truly defined as a SNP, it has to be present in different individuals of the population. It has to be passed down from generation to generation. So, does that mean that if I were to get a mutation from sunlight, let's say I got a bad sunburn, and I carry a mutation in my skin cells that were sunburnt but survived, didn't perform apoptosis, is that new mutation derived from sunshine considered a SNP? Well, no, because it's not being passed in the germline. It's not being passed to other individuals. My skin cells, thankfully, are not going to have sex with other skin cells and make whole new people. That would be wild. Instead, we distinguish between SNPs, polymorphisms that segregate between the population, and variants that exist in particular tissues, single nucleotide variants. Variants are what we study when we study different viral infections or cancerous tissues, simply because they're not being passed down from generation to generation. And I know that a large number of y'all will go into cancer research or somatic cell research. Not everyone here is going to be studying what happens with gametes. But knowing that there's a distinction in the way in which we name them is very important, because you might be looking at some SNVs, single nucleotide variants, that correspond in similar places to SNPs, and you can actually derive some information from them in that regard. Now, we're going to always be looking at these SNPs in terms of what type of SNPs they are. And this, again, cool little infographic from Wikipedia again, shows you the flowchart of your different type of SNPs. We're no longer going to be talking about SNVs in this class, simply because this is a class on genetics, and genetics requires you to pass on from generation to generation. You may take a pathology course or something of the sort in order to better study SNVs. So we've got our 2 types, our non-coding region and our coding regions of our genome, where SNPs can exist. Now, the non-coding region roughly makes up about 97% of all of your DNA. Contrast that with your coding region. If we're estimating it high, we get a number of about 3%. So the vast majority of your genome is going to be non-coding versus coding, meaning that most of your SNPs are going to be found in your non-coding regions. Now, what do we have in terms of targets for these SNPs in our non-coding regions? Well, we can have regulatory regions, some of our promoters, some of our enhancers, some of our other transposable elements that happen to be methylated or demethylated, or regions that can be acetylated, all the stuff that goes into eukaryotic gene control, and repeats. Repetitive sequences that can carry these mutations where they might not have so much of an effect other than telling us who's more related to who. If you remember back to our human population ancestry derived from mitochondrial SNPs, most of the mitochondrial SNPs are present in a repetitive or spacer region in the mitochondrial genome between two functional genes. Mutations can accumulate much faster, or SNPs can accumulate much faster in these repetitive regions that do not code for sequence simply because they may not have an effect. This is fundamentally the reason why we use a lot of these non-coding regions that quote unquote evolve faster, or mutate faster, because they can tolerate more variation. This means that when we look at our coding regions, we can also see variation in the number of SNPs that we have between synonymous and non-synonymous. Because synonymous SNPs, or SNPs that do not change the amino acid codon when being expressed as a protein, because they don't change things, they just change GC or AT bias across the genome, synonymous SNPs are considered to be much better tolerated. Whereas non-synonymous SNPs can be selected for in cases that are very, click the wrong button, that are in cases that are very rare. I'll keep that edit in, I thought it was funny. Or selected against. Because nonsense or missense mutations can oftentimes truncate or destroy the function of the protein, and therefore the selected against is going to be the more common way, and why we'll see variation in terms of the number of synonymous versus non-synonymous SNPs. This is going to be incredibly important when we enter the third unit of this class, our population genetics unit. So I'm pointing it out here. I need you to keep it in mind, but we won't do more with that information up until we start looking at comparative genomics, of looking at different species against one another. For now, when we're talking about our SNPs, we're going to be looking within a population, and therefore looking within a single species. Ultimately, all of these SNPs are identified by comparison to a reference sequence. All organisms on planet Earth that have been sequenced are simplified down to a single reference genome. The reference genome for humans is not even a single reference. It used to be. It used to be the reference genome of a single individual named Craig Venter, who had the ego to say, hey, guess what? I think I figured out how to sequence things faster than the entirety of the United States of America, and maybe I can beat the Human Genome Project at sequencing the human genome. And then he kind of looked like maybe he was with a small team of scientists, and so then the US government and all the public institutions paired up with Craig to publish the first draft of the human genome. From there, we included a random subset of individuals from Buffalo, New York, so probably not the most genetically diverse group of humans. And we actually know that it's not that genetically diverse because we've sequenced lots of humans since then to try to better assemble a reference genome. But even this reference is a composite of organisms in humans. In corn, for example, it is one particular inbred line in corn named B73 that is very, very important to corn breeding history. And while it does not matter which individual you select as the reference genome, or rather, it shouldn't matter a ton, it does matter that you know how many individuals contribute to the reference genome and how much genetic diversity is captured there. Is this individual that you've used as the reference genome, are they a highly differentiated individual, meaning that they're going to have a lot of variation from person to person? Or are you using an individual that is more intermediate, or are you using an ancient skeleton from a long time ago and they're going to have variants that maybe don't exist today? There are lots of different things that you need to consider when talking about SNPs in your reference genome. Now, SNP density varies across the genome because SNP density is a product of a couple of different things. So I'll take a moment here to ask you this question. What are the forces that will determine SNP density in the genome? And I'm looking for the 3 things that are most likely to impact SNP density. Well, first and foremost, how do we make SNPs? Well, mutation rate, right? SNPs are fundamentally mutations. So if we're looking at SNP density in any part of the genome, meaning how SNP density on the long arm of chromosome 3 compared to the short arm of chromosome 6, we're going to see whether there's a variation in mutation rate across those 2 chromosomes. And now, while in many cases we don't assume that there's going to be a difference in mutation rate within a single genome, what we do know is that mutation rate can vary slightly based on the species that you're looking at. So, SNP density can be a product of the mutation rate of the species. Now, within a genome, so I'll write that. This can be a cross-species variation. Now, SNP density can be a product of another force within a genome. Something that varies based on chromosome or region of the chromosome. And that is the all-important recombination. Regions with high recombination oftentimes experience higher mutation rates, plus they can mix and match their different haplotypes, allowing them to bring mutations that have occurred on one haplotype into another haplotype. And then when you study that particular individual, you'll see that they carry these SNPs. So recombination can impact SNP density by bringing together more mutations that have occurred on other haplotypes into a single individual. Last but not least, SNP density is a product of selection. And this is where we saw the difference between our types of SNPs, where we see non-coding regions, where there's going to be the vast majority of the DNA, and an even higher proportion of the SNPs. And that's important to understand. Know that you won't get the same fraction of SNPs within your coding and non-coding regions. Yes, your non-coding regions make up more of your genome, but they also make up an even higher percentage of your SNP density compared to what we see in coding regions. And that's, again, because of this selection against a huge portion of SNPs. I mean, looking back at this flowchart, synonymous SNPs can be tolerated in many cases, but all of the non-synonymous types, the missense, the nonsense, the truncations, etc., they will likely not be tolerated and selected against. And even in cases where some new mutation can be beneficial to the individual, we'll spend the last portion of the class talking about how positive selection can actually reduce SNP density in a localized area. So selection in any form, whether it's positive or negative, can impact SNP density. Now it's also kind of cool that SNP density can be predicted by certain phenotypes of the DNA that you're looking at. Take a moment to think if you can see what kind of genetic features would predict SNP density based on what you know about regions of the genome and how they might differ. Well, the most straightforward thing that can predict SNP density is the presence of genes or not, right? If we see that genic regions or coding regions have lower SNP density, then whatever molecular features of our genes can also predict SNP density to be lower. But this isn't just true based on coding and non-coding, it's also true based on the type of bases that are present. Regions high in AT content, adenine or thymine content, regions that are microsatellite repeats, not even confident I spelled microsatellite correctly, but we'll talk a little bit more about what microsats are in a moment here, but regions that are repetitive with a AT motif are going to have a higher SNP density or a different SNP density than regions with higher GC content. Now, what's the mechanism behind this? Well, you may remember when we draw G to C and we draw A to T, how many hydrogen bonds are between them? 3 between G and C and 2 between A and T. What this means is that regions that are low in GC content are going to be less stable, right? The more AT you have, the fewer hydrogen bonds you have connecting your two strands of DNA, and therefore, the more quote-unquote unstable that DNA is to things like heat. Now, when you have high GC content, it means that you are in a genic region. So between these molecular forces about stability and these forces about genic content of AT versus GC bias, so your higher GC in your genic regions, we see that SNP density can actually be predicted by AT content. Kind of neat, huh? Now, GC content and AT content vary by species. And this is kind of a hint that we need to start looking outside of a single species or even outside of a single population to better understand how these SNPs look and how these SNPs segregate. Because some of our variations, some of our SNPs are going to be really old. You can imagine that there are SNPs that segregated in wolves before the subset of wolves that were domesticated into dogs, a subpopulation of wolves that no longer exists on planet Earth today, at least as of recording this lecture. That is the current best evidence of our information about wolf populations. The subpopulation of wolves that over generations of breeding are our dogs no longer exist on the planet. But some of the SNPs that were present in that population of wolves are carried forth into the modern day population of dogs. This means that some of these SNPs may be really, 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 really old and held in common between dogs and wolves, and some of these SNPs will be more recent, being defining features that differentiate different breeds of dogs rather than informing anything about wolves. Wolves may not have a SNP in a place that differentiates a husky from a boxer. Now, this GC content can vary across species. We see it go in humans, having an average of 41% across the entire genome. In yeast, the unicellular Ascomycete, having a content of 38%. And remember, when we're talking about humans and yeast and these other species, Arabidopsis thaliana, the model species for plants, keep in mind that tree of life. We see that Arabidopsis thaliana has an even lower GC content at 36%. Now, even across the human genome, we see variation within regions or within chromosomes, within 100,000 base pair fragments, where the human genome can find regions that are as low as 35% GC and as high as 60% GC. That's a huge amount of variation. That means that we'll have regions that simply because of their higher GC content may have varying amounts of SNPs, which we're going to take a look at in a sec. But I want to wrap up this discussion of variation in GC content across species to discuss something that touches back to phenotype, because everything that I've talked about here has kind of been really detached from any kind of molecular manifestation, right? So we look at the DNA, how does it actually impact what we're looking at? How do I convince you that all of the things that I'm talking about here don't just matter for molecules of DNA, but rather for the overall survival and life habits of these different organisms? Well, we see that different mammals have different amounts of GC as well. Just like we see variation across different branches of the tree of life, we see variation within mammals. In fact, there is variation within mammals to an extent where some have more GC in their genome than AT. Things like shrews, the relatives of elephants, that are part of our Afrotherian branch. We also see particular phenotypes connecting to this, where genome size decreases with increasing GC content, and body size decreases with higher GC content. Sorry for the sloppy handwriting here. I'm trying to slam in all this interesting information. Maybe didn't leave myself enough space. But I do find that that's very, very interesting and something that you can pursue when trying to study different organisms on planet Earth. Some of you who want to go into careers in zoology or veterinary medicine, understanding some of the genetics and features of the genome that you're studying are very important. And especially when it comes to trends in terms of how they adapt to their environments. We see evidence of that high GC content and body size, particularly in the shrew. But I can also link to you the paper that discusses this variation across different species. It's quite interesting and was published in the not too distant past. I believe it was in sometime in the 2010s that this look at GC content across species was first published. So when we're talking about SNP density, because obviously this tangent about prediction of SNP density with AT content matters because we want to look at SNP density across the human genome. So here we have it. Here's our chart of SNP density across the human genome. You could pause the video here and stare at this a bit yourself, or you could go to Wikipedia on SNPs and check out this table for yourself. That's where I've poached it from, as with most of my infographics and other materials. You'll notice a couple of things when looking at this table. You'll notice that a number of thousands of bases per SNP varies across the genome in terms of chromosome, but stays kind of steady at around 3,000 bases per SNP in the human genome. Each of the chromosomes has something about. This is the chart that we're looking. TSC SNPs are a different type of SNP, so my bad, not there. You're looking at about 2 SNPs per 1,000 bases, or 1 SNP per every 2,000 bases, and then you're looking at the total number of SNPs per chromosome here. So you see variation in chromosome size. The thing of note here is to take a look at the SNP density on the X and Y chromosomes, those for which recombination rates are suppressed. If recombination rates are suppressed, like between the X and the Y, you actually get fewer numbers of SNPs. So it's more kbs per SNP. Again, because recombination rate plays a huge role in SNP density. Now we also want to connect, coming full circle here, SNP density, recombination rate, and the genetic map. So when we look at the genetic map of humans, we see that one map unit is roughly equivalent to 1 million base pairs, or megabase pairs. This varies across different species, but it's about 1 to 1 million, so you get one recombination event per meiosis per every million bases that you travel across the genome. Now this varies drastically because centromeres have very low recombination. Oops, we'll just use rho, whereas the chromosome arms have very high recombination. We also see something absolutely fascinating. I didn't learn this in grad school simply because I studied plants, but a fair bit of reading after grad school taught me that you actually get variation in recombination rate by gender. So you get variation by region, but also variation by gender. And this is because of the way in which eggs and sperm are performing their meioses, performing these things in terms of how much recombination is taking place. When looking across the human genome, or the human male genome, we see that males have a genome that's approximately 2,500 map units big, and females have a genome that's approximately 4,500 map units big. Meaning that there's more recombination on average in females than there are in males. Kind of interesting, right? Now, it's important to note that we have different recombination rates across our chromosomes, and ultimately both genders combine and switch, whether they're in a male or female, based on the generation, because so many of our chromosomes are autosomes rather than sex chromosomes. So it's important to understand both the average when looking across males and females, but then also understand the differences between our genders. Now, one topic that is mentioned in your book that we're not going to take a deep dive on is something called the map function. Now, the map function is the way to correlate your recombination to physical distance. Now, unless you go into this field where you're looking at recombination rates as they vary across structural genetics, I don't think this is going to have a wide application to most folks, so we won't spend the time in this class going over it. Your book does a medium job of explaining it, so if you want to take a deep dive, I'd certainly recommend that subsection in your text. Now, the last topic that we're going to discuss today is some of the constituents of that non-coding region of your genome, where you're going to find a huge portion of your markers and the type of markers that exist in those areas. I'm talking specifically here about repetitive DNA. Now, drawing back on our previous lectures, you know that repetitive DNA can be of two broad classes: our long repeats like transposable elements, or the ones that we're going to focus on here, our short repeats. Short repeats, like tandem repeats or microsatellites. Here's the aside where we talk about microsatellites, because microsatellites were named because they formed a giant block on a gel electrophoresis which is like that game of Plinko, where sugars can act as molecules that slow down the progression of DNA down the sugar block, and you get size separation of DNA. Now, when we cut up DNA using restriction enzymes, which we'll go over in a second, we saw that regions that have a lot of SNPs cut up to many small fragments. Regions that have lots of variation and can be accessed by these specific pattern-seeking restriction enzymes cut up to be very small. And then there were huge chunks of the genome that stayed floating above the rest. Now, we thought, no, actually, I don't know what the scientists thought the first time that they were looking at them as to what exactly separates those regions. But we do know now that they are made up of these incredibly long repeats where we see just patterns repeated one after the other. No genes, just structural DNA. Now we separate our microsatellites, repeats that are less than 10 base pairs long, meaning that you might get an AATTTT, AATTTT for something like 100,000 letters in a row, kind of wild, or mini-sats, repeats that are a bit longer, 15 letters long, up to 100 base pairs long, and then those will repeat. Above our mini-satellite repeats, you might get some of our, what would have been called, in my world of literature at least, the tandem repeats. These are things like centromeric repeats that have lengths of 150 to 180 base pairs. And that length for those repeats is important simply because that's the length needed to wrap around a centromeric histone. So there can be structural reasons as to the particular lengths of these repeats. Now, this repetitive DNA can be used as an excellent molecular marker, not because we're going to try to find SNPs in them, which is why we're talking about repetitive DNA separate from SNP density, but repetitive DNA can be used as a molecular marker because of copy number variation. We can get simple sequence length polymorphism, like what we talked about when we discussed Fragile X syndrome in humans, where someone with a small number of microsatellite repeats, this was a 3 base pair tandem repeat of CGG, CGG, CGG. Now, if you had just a couple of them, something like 5, you were not at risk for Fragile X. If this had copied itself on the order of something like 20 of them, then you were pre-Fragile X. And if you had something like 200 of them, this would indicate that you have the condition. And by looking at the copy numbers of tandem repeats through processes like gel electrophoresis, we can genotype individuals not for SNPs, but for copy number variants. This is not to be confused with another topic that we may explore in some of our labs of copy number variants of particular genes. This is copy number variation in a single region of a particular repeat. Now, gel electrophoresis, as a reminder, works because DNA is charged. You're going to put DNA in a well at the top of the gel next to the negative charged part of the gel box. Then you're going to run a current across this, and it's going to run towards the positive side. This is what a gel box looks like. Ours in the classroom are actually teal colored rather than yellow colored here. And then you're going to get a banding pattern or a DNA fingerprint. So how actually is this DNA fingerprint established in tandem repetitive sequence? Well, going from the entire process, we see DNA is extracted. In part 2, you're going to then do some sort of molecular biology to the DNA. You may amplify it by throwing in particular PCR primers and amplify particular regions of the genome that you're interested in. Or you may expose it to a restriction enzyme that's going to cut restriction fragment length polymorphisms to create these patterns in a genome-wide fashion. Once that DNA is processed, you're then going to add it to the wells on this gel. The electric current is going to be applied, negative on the side of the wells towards the positive because DNA is negatively charged. It's going to run towards the positive. If you flip this around, you will then run your DNA off of your gel, and you'll wonder why you have no DNA in your gel. The DNA is going to be separated inside of your gel based on size. Now, DNA is invisible to the eye unless you stain it. So part 6 is going to be the staining of the gel with some sort of fluorescent molecule. Now, I'd imagine by this point in the class, you are familiar with this, since we've hopefully run a couple of gels if everything's going well. Of course, always dangerous to include in recorded material, but I'm fairly confident we've got it done by this point. And so this should all be some sort of review and connection to how SNPs and repetitive differences in genomes can be used to make molecular maps. We can use gels in pedigrees, where you can identify particular banding patterns to genotype progeny. Boy, wouldn't that make for a cool problem. And we can also see some really cool patterns, like in this gel, looking at what happens in terms of bacterial diversity in the gut before and, grab highlighter here, before and after bombing it out with antibiotics. So both of these gels are of two different individuals where you see patterns before and after they've been bombed out with antibiotics. You'll see that there is a high amount of diversity of bacteria before, and then we see after the exposure, we see far, far less diversity of bacteria in both individuals. And now this allows us to tell these patterns and diversity of organisms present based on these gel electrophoresis patterns. In the same way, we can establish these DNA fingerprints that are unique per individual because we're looking at a genome-wide pattern. This is also what's used commonly in forensics to establish whether the DNA on the crime scene is tied to an individual. So, this last image here you see that there is a relationship in terms of how far a fragment goes, the distance migrated, and its molecular size. The smaller the fragment, the farther it goes. The larger the fragment, the uh, less it travels. So big stuff at top, small stuff on bottom. Just moves quicker, like through the Plinko board. So that's where we'll wrap things up in terms of our connection between molecular markers and mapping. Please do make sure that you have a good grasp on our tri-point crosses, 3-point crosses, as they will certainly feature prominently in what you need to know in this class. Again, whether you take it here or literally at any other institution in the world. I'll see y'all next time.